Welcome everyone to our Cancer Genomics Cloud webinar series. I am Sarah Fisher, and I am one of our presenters today. I am a community engagement manager for the Cancer Genomics Cloud, and I have a PhD in ecology and evolutionary biology. I want to also introduce my co-presenter, Jared Rosowski, who has a PhD in biomedical engineering from the University of Florida. Jared works on a sister platform, Cavatica, and is here today to talk to us about GA4GH DRS, Data Repository Service, and a new workflow on the CGC. So with no further ado, go ahead and take it away.
Awesome, thanks so much. Give me a second to share my screen and get this underway. All right, hopefully you can see nice Valsera logo there. If I go into presenter mode, I just get a thumbs up if that looks good to you. Awesome, thanks so much. First of all, thank you everyone for attending. It's great to see people in person. I know this is recorded and you can watch it later, but it's definitely engaging to see some faces or see some names at least on the Zoom call. I'm super excited to be here. I've been listening to a lot of podcasts, so I'm hoping this kind of kicks off my podcast career. I've been a part of this project for a little while now, so I'm really happy to be presenting where we are today. One of our missions at Valsera is to promote fair data principles. And if you're not familiar with that, you can see it on the slide, but it's findable, accessible, interoperable, and reusable. And that's referring to data within the ecosystem. But in simple terms, what we're trying to do is allow researchers to access the data that they need to do research. Today, we're going to be focused on the interoperability between the Sequence Read Archive, or SRA, and the Cancer Genomics Cloud, and by extension, all of Valsera platforms. The National Center for Biotechnology Information, or NCBI, at the National Library of Medicine hosts and manages the database of genotypes and phenotypes, or as pretty much everyone refers to it, as dbGaP. And they also host and manage the NIH's Sequence Read Archive, or SRA. dbGaP provides and manages access to protected data related to human studies that have investigated the interaction of genotypes and phenotypes. SRA is the largest archive for public and controlled access next-gen sequencing data. We're all aware of the not-so-new NIH guidelines on data management and sharing, and the SRA is a generalist repository for sequencing data. I'm assuming, and I'm sure most of you know what the Sequence Read Archive is, since you're here today to hear about it. But just to give a little bit of data and insight into how much data and what's there, the NCI Genomic Data Commons, which is integrated very closely with the Cancer Genomics Cloud, has high-quality data sets related to cancer. Specifically, there's the TCGA, or Cancer Genome Atlas, Human Cancer Models Initiative, Foundation Medicine, and a variety of others. And in total, that data adds up to about 8.8 petabytes of data. Compare that to what's in SRA. This is a number that was projected back in 2019, since I couldn't find current data. But they said by 2023, there would be about 33 petabytes of data in SRA. And I just want to note that that's compressed data, so it's not exactly apples to apples. Using a very conservative approximation of 2-to-1 compression, we could say there's probably 66 plus petabytes of data in SRA. So relative to the NCI Genomic Data Commons, a huge amount of data is made available to you. And what we're presenting today is that now all of that data within the SRA is available to you to use on the CGC. One thing I want to say is we talk about petabytes of data. I can't really fathom what 66 petabytes of data is. And to be frank, I don't care too much because I'm not going to use all 66 petabytes at one time. What I want to know as a scientist is the data that I want to analyze in there. And I can't say unequivocally yes, but I can say most likely yes. They have data from many different species, human and not human, many diseases, many sample types and tissue types. All of the sequencing data you can imagine in a generalist repository. So what connects the CGC and the SRA in general? Both of these programs are part of the NIH National Cloud Platform Interoperability Program, or NCPI. And this is a collaborative project between NIH and external partners seeking to create a federated genomic data analysis ecosystem. We focus on interoperability, and the NCPI program is ensuring that researchers can both find and integrate data more easily from each of the participating programs. One thing I want to point out, you may have noticed this already, is that Valsera is a big part of 3 of these partner systems. There's the Cancer Genomics Cloud as part of the NCI, Biodata Catalyst, and we are tightly integrated with Kids First and Cavatica, if you're familiar with the Cavatica platform. I would be remiss, since we're talking about interoperability, if I didn't also bring up the GA4GH, or Global Alliance for Genomics and Health. It's a policy framing and technical standard organization seeking to enable responsible genomic data sharing. There's many different aspects of this, so I'm not going to get too deep into GA4GH today, but it's actually a very opportune time to be presenting because almost exactly a year ago, our fearless director of interoperability, Michele Mattione, presented all about GA4GH alongside Angela Page, and I stuck a link there. I don't know if we can do one of these like links pop up, but later on you'll be able to click that link. You can also go to the CGC website and find that link, and you can watch that webinar. Today, we're going to be talking about one of the standards specifically that came out of GA4GH, and that's the Data Repository Service, or DRS. The definition, or one of the definitions there, is it's a standardized set of access methods that are agnostic to cloud infrastructure. But if I had to simplify that down, it's really a way to allow symbolic linking to the data that's stored in the cloud. And what that means is that the data gets stored once, and we as researchers, specifically using CGC or any of our platforms, can link to it. It means we don't have to pay storage. That data stays where it lives. The data owner pays storage, and we can use it. So as a researcher, that means that I can access the data, I can save time, and I can save money. And I think those are really important products of the DRS standard. Now, circling back to SRA, the old method of accessing data from the SRA involved downloading data from the SRA either to your local on-prem storage or to a cloud bucket that you can store. And that worked. It worked okay, but it means that you as a researcher are now paying for the storage of that data. And in big picture, now there's 2 copies or multiple copies of that data and multiple people paying storage for it. We, as Valsera, have integrated tools within the CGC that allow you to use that workflow and download data directly into your project. So you can streamline that process. But again, we're not using the latest in interoperability standards. So if I go forward, out with the old, in with the new, the SRA data is now on the cloud and available via DRS. This is a screenshot from an article back in 2020, but really just showing that the data is now all available in the cloud. But that doesn't necessarily mean that it was available via DRS until now. So we have implemented a tool, or worked with the NCBI to implement this tool that allows you to get DRS links for data in the SRA. In other words, you can symbolically link to data in the SRA so that you don't have to pay storage costs. The files will show up in your project as if it's a normal file, and you can run your analysis, and I'd say it just works. There are some important notes, and again, I want to reiterate that the user is not charged for storage since it's symbolic linking. Any of our platforms, including CGC, can import data from the SRA if it's open. I want to give the caveat that control data works only on Cavatica thanks to the RAS integration, but it is possible using the Valsera's interoperability stack to bring that data from Cavatica to CGC. And I'll put a plug in here for office hours. I'm sure you all know about it, but if you have questions on how to do this, Sarah and our other community engagement managers are happy to help. It's a fairly simple process. The other caveat that I'll give is that currently this is working only for data that's in hot storage. Hot storage essentially means it's readily available and you can access it immediately. I'm going to talk a little bit more about that, so I don't want to get too deep into it just yet. But here we are, current state versus future state. We designed this tool to import the raw FASTQ files, generally thinking that that's what most people want because that's what we're used to. Currently, if the data is in cold storage, you can request data via one of the old mechanisms, and then again, you pay for that storage. However, Sarah did some wonderful investigative work and discovered that most, if not all, of the data is available in hot storage in .sra or .sra Lite files. Now, I've put a link at the bottom of the slide that tells you a bit more about what those are and what that means. But essentially, it means that you can access almost the entirety of the SRA in hot storage in these file types. It makes sense that the NCBI would want to use compressed files in hot storage because the smaller they are, the cheaper the storage is going to cost. If you keep all of the raw files in hot storage, the cost of storage significantly increases. So Sarah will show a demo later on about how we can convert these .sra files and back into FASTQ files and how you can incorporate that into your workflow. But again, I just want to mention current state is raw FASTQ files. I would say in the next week or two, we should have this up where you can access the SRA files and use them. So this, I mean, to me, this is huge. You can access almost all of the SRA through the CGC. Now, I'm going to go through some slides that show how to do this on the platform, but Sarah's going to take over in a few minutes and actually show you live how to do it. I just want to point out a few key things. So one of them is that we have this new workflow on the CGC called SRA to DRS converter. It starts with either a metadata file from the SRA or an accession number if you want the whole study. And it ends with a list of DRS, a file with a list of DRS IDs and DRS URIs. You can then go on our platform and import that file as you would any other GA4GH manifest file. And all of those files are then going to show up in your project. So you can get the metadata file from SRA. It shows up on that website. You can download it and then you use that as input to the workflow. Or you can use, if you know the accession number, you want everything there. Oh, sorry, this would be showing the input. Next slide. This shows the accession number if that's what you want to use. The output then shows up here. Specifically on the right-hand side, you can see the updated SRA table includes those DRS URIs that you can use to import files. I think I'm getting a little bit ahead of myself, but we'll catch up. The output, if you take a look, has a column called is online, and that tells you whether or not the FASTQ file you're interested is in hot storage. Is online is true, it's hot storage. Is online false, is in cold storage, means you cannot currently access the file as it is. The DRS URI is really that symbolic link. It's the pointer to where the file lives, and that's what our platform uses to import the files into your project. Now, the downstream of that is once they're in your project, I assume most of you have run a workflow on the platform before. If not, no worries. It's pretty easy to do. As an input file, you just select the file from your project. And what this slide shows is that you can use a DRS file, or you can use something that you've uploaded and is stored within the project, and it works the same way. So you can have files from the SRA. If you want to combine them with your own files that you've added to the project, works the same way, no difference. And so just to wrap up, because I think there's a few key messages here, we have unlocked the data from the SRA, and you can now use it within the CGC. This saves time, it saves money, and it means you can access a whole heap of data. I want to make some acknowledgements before I turn it back over to Sarah. The NCI funded this effort, so we want to thank them. We want to thank the NCBI for setting up the DRS server and working with us to interoperate. Our bioinformatics team put together this workflow. Divya Sain is the program manager for the CGC. The interoperability team, which I'm on, also includes Michele Mattioni and Surya Saha. We have our great product team led by Milos Trebojevic, and then our wonderful co-host, Sarah Fisher. So I will stop sharing and turn it over to you.
Thanks so much, Jared. So I'm going to just rearrange my windows for a second here, because what would a webinar be without a host struggling with their windows for a second? But now I'm ready. So I'm going to go ahead and do a live demo of the new workflow. And you should be seeing my screen. I am logged into the Cancer Genomics Cloud, and I'm on my project page for the webinar demo. The first thing I want to show you is how you actually find this workflow. So from public apps in workflows and tools, when you browse apps, you can actually simply search for SRA, and you'll find all of the different tools that we have available for working with SRA data. SRA to DRS converter is the one that we are interested in right now, but don't sleep on SRA download and set metadata. It's also a pretty nice tool. It does involve actually downloading and storing the FASTQ files in your own space. Right now, we're interested in how do we access files without actually having to download them. So let's go back to my project where I have copied this app into my project already. So as most of you are aware, when you run an app, you go to the apps page, find the one you want to run, and you'll click run to start a draft task. And as Jared mentioned, there's a couple of places where you can provide inputs for this workflow. I really like the fact that you can just give it an accession number, including one of the bioproject PRJNA numbers, meaning you can just see, like any bioproject, what's actually available in hot storage that I can use right now. But I want to talk a little bit more about using an SRA metadata file, because I think that that's probably the use case that most people are going to be working with. So I'll switch tabs here to the SRA run selector. I have entered in a PRJNA number for one of the 1,000 genomes subprojects. And I've already filtered my runs down because the total here is 1,000 runs. I don't want to work with that much data, even if it is all hot storage. So I've filtered by assay type, whole exome sequencing. I've filtered to the Yoruba population, and I'm just using the BI sequencing center. That gives us 33 items. So I'm going to select all 33 of them. And now I have a toggle here. I can turn selected on here, which means that when I download the metadata file, it's the metadata file that we're going to be using, it will only include these accessions. So I will save that file. I actually already have saved it. And then we can, from there, go to the draft task. I have already uploaded this metadata file, so I'm going to select it from my files here. And it's actually going to be using this one. We can take a look at this and just see what information is actually included in this file. It's a normal SRA run table. It has all of that metadata that we saw on the run selector. But most importantly, it has the accession numbers here. So that's going to be our input in this draft task. And then there's really no need to change any of the other parameters. You can, if you wish, increase the number of splits And that can sometimes make things run a little bit faster because it'll split a long table into a few chunks. But this is only 15 samples, so we're not going to have to worry about that. I am not going to run this right now because it'll take a little bit to actually finish. But Jared had shown you some of the way that the output looks. This is the one that I want. And here is that finished run using that same SRA run table. Let me close this. That's not helpful. So this is basically the log file that shows what the script did to find the DRS URIs. This is only interesting if you are really into interoperability, but if you are, this might be pretty interesting. What we're really interested in is this hot.csv. That would tell us if any of these files are available in hot storage. And I am sorry to tell you that none of them were. This is sort of a disappointment, of course, because I was hoping that they would be. But one thing that is kind of interesting about this workflow is, as far as we know, this is one of the only ways to find out if the original FASTQ files are actually available on hot storage. But never fear, we are using our normal cooking show modality of run some things first and make sure they work. And I do have live DRS URIs to show you. This is one from a bioproject 489753. And in the hot CSV file, it does have several DRS URIs for FASTQ files that are available via DRS. So I'm actually going to think that I tried these earlier, but oh, they're all there. I'm going to actually delete these to show you so that I can show you how the DRS import really works. So delete that folder also. So I have these DRS URIs. I have that whole spreadsheet here in Excel, and I'm just going to copy these URIs, and I'm going to go to add files, and we'll use the GA4GH data repository service. You can actually use the hot.csv file as a manifest file, but it is a little bit quicker to just paste the DRS URIs in. And I'll put a tag live demo on these. And since this is 14 files, it might take a second. But if anybody has ever actually worked with the SRA toolkit on the command line, you know that actually dumping your FASTQ files out to your local storage is both very time consuming. It just takes a long time, and also a fairly frustrating process because the SRA toolkit will sometimes lose its connection and have to restart. It's a pretty graceful toolkit. It'll restart at the same point that it left off, but it can be very frustrating if you're having to manually manage those downloads. Here we gave the DRS URIs, and it is, of course, going to take much longer than I want it to for a live demo. But in reality, it'll only be 45 seconds to a minute. While I'm waiting on that, what I do want to show you is what Jared was hinting at the end of his talk. We mentioned that the DRS connection works for files that are in hot storage. Quite understandably, NCBI does not keep most of the FASTQ files or CRAM files in hot storage. Those are large files. And the SRA and SRA Lite formats are much more ideal in terms of compression and their storage footprint. So what we really would like then is to just be able to connect to those .sra files. One of the, oh, look, they're all here. That didn't take all that long at all, even if I was a little bit impatient about it. It's funny how when you start working on the cloud and you start like using this kind of technology, you start to get impatient about seconds of time. Whereas when I was doing my dissertation and I was downloading a ton of things from SRA to do this meta-analysis, I spent probably a week or so just getting the files downloaded before I could even process them. So perspective for myself. What I was going to talk about is one of these apps that we have in our public apps. When you search for SRA, you will find this app, SBG Convert SRA/BAM to FASTQ. So this app actually does a couple of things. We only really need one of its functionalities, but it will take an SRA format file and use the SRA toolkit FasterQ dump to decompress that to the FASTQ file. This is exactly what SRA toolkit does. This is in fact SRA toolkit. So what that means is if we have files that are live via DRS and our SRA format, we can actually use this app to convert the SRA to FASTQ and plug it into our workflows. Let me show you what I mean. So here in my files, I have an SRA format file that is connected via DRS. So we have this little bucket here, and we can see that the DRS URI is the ncbi.nlm.nih.gov. So this is NCBI's DRS server. And it's in SRA format, not a plain text format, as we can see if we try to look at the raw view. I'm not even sure it will show us anything. Yeah, it won't even show us anything. So now I have this symbolically linked. I'm not paying any storage for it, which is good because it was, you know, even compressed, it's 3 gigs. And just this morning, I was able to create a new workflow that uses that DRS connected SRA file. So I called it SRA streaming demo because we are actually streaming the SRA file into a workflow. And I'm going to go into the tool editor to show you how this is put together. So first, I'm using this app that I mentioned, SBG Convert SRA BAM to FASTQ. I have this input here where I'm going to put my DRS connected SRA file. And then that's going to be processed by this, and FASTQ files will be created. But I'm not actually going to even worry about saving those FASTQ files. One of the things that we often stumble on, or I often stumble on when using cloud connected tools, is doing everything stepwise and having these derived files. But I'm not really interested in having the FASTQ files. I want to analyze those files down, pass them through a salmon workflow to get transcript level quantification and gene level quantification, right? I really just want to quantify these RNA-seq files. And so I don't need to ever have the FASTQ files. The FASTQ files will be created from that SRA file, and then they'll be directly streamed into the salmon workflow. And then from salmon, we will get out our quantification. We can also at the same time stream them through FastQC in order to get our reports about, yes, our reports about the FastQ quality. So that's what I created this morning. And again, the basic gist here is any workflow on the platform that has FastQ read files as an input could have this SVG convert SRA to FastQ as the workflow that pipes in those FastQ files. You can basically just put this guy in front of any workflow that takes a FastQ, and we can directly stream the SRA files into our workflows. So let me just show you that this actually works, because I was very excited that it actually worked. Here we have my SRA streaming demo. The input file here is that DRS connected SRA file. And then we also have to put in our reference files for the salmon transcript abundance workflow. So we have our Ensemble 95 gene annotation file. We have our H38 genome assembly. And we have a transcriptome, the Ensemble transcriptome FASTA, for the salmon workflow to use as a reference. And the outputs were exactly what we wanted. We're really only putting one run in here. So when we click on this expression matrix to see the number of the number of reads for each gene, we'll get a bunch that have 0. But if you sort this way, we can see that we have our genes and our counts, which is all you need to start doing your analysis. And the take-home message from this is we have created a 1 megabyte file that has the information that we need for our analysis by streaming this 3.8 gigabyte file through this workflow. This says that this took less than a minute because I copied it from another project, but it took 2 minutes. It cost $1.30 to, instead of downloading these files, storing them, and then converting them to counts, to just stream directly from the SRA file live on a DRS server and get the counts out of it. I was very excited about this, in part because I have files from my PhD dissertation that are in SRA that I would not mind being able to reanalyze. But it's also really exciting because it means that anything that is on the SRA is now accessible by our workflows. Part of the value of this is that we're trying to move more towards this analysis to data paradigm. We often say we should analyze the data where it lives. And that's because the data just keeps getting bigger and bigger. Not just that there's more of it, but the files that come off of a nanopore machine are much larger files than the files that come off of an Illumina machine, some of the Illumina machine. So it's starting to just not make as much sense to move the data around. It makes a lot more sense to just put the software that we're going to use on the machine where the data is and analyze the data. It's faster, it saves computation time, it saves download costs, but I think it also really saves a lot of researcher time. I know I have spent many hours of my life just handling FASTQ files. We don't have to live like that anymore. And that's kind of where I'm at with this. I'm trying to remember if there's anything else that I wanted to make sure that I showed you, but I think I've shown you what I wanted to. I'm going to stop sharing and we will, I see there are maybe a couple of things in chat. Oh, okay, yeah. So let's go ahead and have question and discussion. Jared, I'm going to ask you a question from Alejandro.
Yeah,
What about the handling of control data? Is it possible to manage it effectively? I'm interested in a couple of studies from dbGaP to which we have access, and we have to download them with the SRA toolkit.
absolutely. So I kind of glossed over it a little bit in the presentation, but you most definitely can do controlled data. There is a caveat. You have to use the Cavatica platform to do the import from SRA. Cavatica currently has RAS integration, which allows the authorization of controlled access files. And then once those files are in your Cavatica project, you can use the interoperability stack that Valsera has to essentially move or symbolically link the files that are in Cavatica into the CGC. I'm happy if we have time and want to, I can show in like 30 seconds how that might work. Otherwise, we can, show you in office hours and take the time we need. I'll defer to Sarah.
I had the same thought. Do you, does
Yeah,
anybody
I can
else
do
have
it.
any burning questions? Otherwise, I think it might be really worthwhile to just go ahead and show that.
All right. I think 8-second rule
Yes,
is
good
up.
job.
So let's go back to this one. So if you're familiar with the CGC, this should look very familiar to you. I can go back to my very messy homepage with lots of files on it. But Cavatica and the CGC are functionally, 99% the same, maybe some minor differences.
One important difference is this RAS integration.
If I go to my SRA project and go to files, I'm just going to do this for one set of files here. So I have these two files with DRS. I've already gone and made a manifest, but I'll just go back here. If you highlight this and then go into the little 3 dots over here and do a generate metadata manifest.
DRS manifest.
Generate DRS manifest. Thank you, Sarah. It put this DRS manifest in this project. So what I can do, I'll just show you what it looks like. If I open it up, these are now on the Cavatica DRS server, but actually they're just symbolic link to the NCBI DRS server. So it allows you to kind of use Cavatica as a pass-through in a sense. And if you go over here to download, it's going to download this file. And then I got to move the zoom bar so I can see up here. If I go over to my CGC project, again, I'll kind of go to the home page just to ground everyone. I can go to files, add files, GA4GH DRS manifest. And then from a manifest file, I can go here. Tell me if I'm going too fast. I'll go to downloads. And this looks like the right one that I downloaded a minute ago. So I'll select that. Check. Submit. It should be fairly quickly with only 2 files.
It'll just take a second. But seconds feel like a really long time when you're doing live demos.
Yeah, when you're waiting and everyone's just looking at you. This is a cool, fairly new feature we have that gives you updates, more detailed updates, I should say, on how the import is working.
Mhm.
The one thing I didn't check was that my Kvatica and CGC accounts are linked, which is a
Yeah.
requirement.
Why don't you go ahead and show that?
Okay. So if I go over here to account settings and go to dataset access, it's going to show which datasets I have access to and which servers I have. So I've logged in through ERA Commons, which if I had access to TCA, TCGA, or these datasets, it would show up as green. I don't, so they show up as red, but you can see my ERA Commons. And then there's some other data repositories you can link to. The
You are
important one is this one, Kvatica. It says I'm connected. So that should be all set. That allows me to read files from Kvatica. And if I go back, fingers crossed, still going, still going.
It's still going.
Um,
I think the upshot here is,
oh, there we go.
yay.
So now you can see in the project, oh, still going.
Yeah, still getting them.
So maybe you want to take my word for it, maybe not, but those files will show up here once this has finished the import.
The upshot here is Kvatica is the platform that has the researcher authentication service enabled. And because it has that RAS integration, you can make an account on Kvatica using your ERA Commons, connect your Kvatica and CGC accounts, and use your authentication on Kvatica to give yourself a tunnel to the files. So we also had a question in chat about recourse for when the files are not in hot storage. And I definitely can talk about that. Let me open up my, wait, I should probably share my screen before I start talking about it. So one of the apps that I mentioned, and I think I may have it in this, is, yeah, I don't have it. Let's go get it. Your recourse right this second, if the FASTQ files are not in hot storage, but are in cold storage, is to use this tool, SRA download and set metadata. So this unfortunately does mean that you are downloading. But okay, yeah, there we go. So we take this SRA download and set metadata. I gotta move this around so I can see here. And this again will just take a run table file. So I can give it this 1,000 genomes run table file. And it's going to run the SRA toolkit and actually attach metadata to what it downloads. The catch here is that when you have downloaded the FASTQ files, now they're in your storage and you're paying for it from either your pilot funds or your grant funds, if that's what you have connected to your CGC projects. However, you could also do exactly the same thing with this that I showed with the SRA streaming app. In fact, I'm going to make a little copy of this. When you make a copy of an app in the same project, you can't have the same URL. So I'm going to add 2 to this. And let's go in and edit it. So just so I know what I'm doing, I will change the title. And then here, instead of using this SRA convert, SVG convert SRA to band to FASTQ, we will actually pop in SRA download and set metadata. Pull out that port. That's where we'll put our metadata file and put our out reads directly into this FASTQ read files. And if we want them, we can output that metadata file again, and we can have a download log. So that's all you would need to do. And this app would still work very much the same. The only difference here for an there is no difference here for an end user. You would still not end up paying any data storage costs, because you would never actually be storing those FASTQ files. You're dumping the FASTQ files directly into this workflow. I think the main difference is that NCBI ends up paying some small amount for data egress. So in terms of infrastructure for all, it's less ideal, but it's pretty nice for an end user. Change to dump. It's cute. So then we also had another question. Do applications require modification to work with DRS files, or is it more like a file system capability that translates read requests from the application to cloud-based requests? That's a great question. Applications do not require any modification to work with the DRS files. One of the screenshots that Jared showed running FASTQ, and I can actually set it up here again. We can run FASTQ at the same time with both DRS connected files and project stored files. So I can take these DRS files and then also these files that have been downloaded, and run FastQC on this sort of like what looks to us as mixed sets of files, but to the applications, it all looks normal. Great question. Any further questions? Maybe I'm just a big fangirl for the SRA, but I mean, I'm very excited about the massive amount of mouse data that this opens up, right? We don't have tons of non-human data ingested on our platform because we are primarily a human cancer platform. The canine data commons, or the canine genomic commons, is connected. But there's also just a ton of other useful, interesting data that are not accessible through dbGaP. They're accessible through SRA. And especially in terms of like teaching bioinformatics, if you wanted to teach bioinformatics using the platform, that means there's tons of other data sets you could access that don't require doing a dbGaP access request. So I'm excited. I hope it's useful for the community.